have a wonderful set of speakers and lots of really, really awesome, great success stories, great examples of case studies, and different options for how to recover species and how to think strategically. So I'm going to start with a quick Reverse the Red video to get you really excited, and then I'll turn it over and we'll do a quick intro and have our panelists do their presentations. Throughout the day today, if you have any questions, please put them in the Q&A function at the bottom. And we'll get to those at the end. After all of the presentations, we'll have some time for questions and discussion.
In a powerful show of support for nature, nearly every government in the world united behind the CBD's sweeping biodiversity plan for life on Earth. They committed to halting the extinction of threatened animal, plant, and fungi species. And in an historic first, they also committed to helping those species recover, so that the life we share our planet with can not only survive, but thrive. Making these goals a reality will take all of us working together. Reverse the Red is supporting governments, conservation experts, and communities around the world as they join forces to activate tried and tested tools and tie global efforts to local expertise. All in a coordinated effort to assess, plan, and act for wildlife. Together, we can save species from extinction. Together, we can secure our planet's future. Together, we can reverse the red.
Again, thank you all for coming and really starting this Reverse the Red Day week off with such a great start. I just wanted to thank our executive committee members and our sponsors for making all of our webinars and all of the work of Reverse the Red possible. Before I turn it over to Katie, I also wanted to thank San Diego Zoo Wildlife Alliance for sponsoring our Accelerator Award this past year. And so Katie will tell you a little bit about the work that San Diego Zoo Wildlife Alliance and the Center for Species Survival for Biobanking is doing there. And we'll have Caitlin share a little bit more about the Species Pledge and the Accelerator Award and how you can get involved in Reverse the Red and Reverse the Red Day throughout this week and throughout the rest of the year. So without further ado, Katie, all yours.
Thank you so much, Megan. It is a huge honor to be able to represent San Diego Zoo Wildlife Alliance in the first day of Reverse the Red Day week. And oops, I have to go back to the beginning of my slideshow. And so what we're here to talk about today is the evidence of success in reversing the red. And so this is such an important thing to celebrate because
Katie, I'm so sorry. You have a little window that says continue to share your PowerPoint.
Oh my goodness.
It's okay.
I don't know. I can't get to it. It's like stuck behind my Zoom bar. Oh, I can move the Zoom bar.
Perfect.
Well, here we are. Great to see you guys. Is everything well with Megan with the presentation?
Yes, looks great.
Perfect. Okay, so species recovery is happening all over the world, and that's not by accident. For every story that we'll hear about species reversing a trend towards extinction, this represents years of partnership, investment, and scientific achievement. So all of the actions in the recovery chain, they range from extracting DNA into the lab, propagators learning how to grow cuttings on agar, local communities engaging in monitoring and threat reduction, and wildlife care specialists preparing animals for release. To the most important part, that can be government partners finding the resources and pathways to enable recovery. And so what Reverse the Red does in celebrating these achievements allows us to publicize models for recovery that can work across taxa and regions, and also builds goodwill for conservation that can validate all of the investment that is truly needed to save more species. And at San Diego Zoo Wildlife Alliance, our mission strongly aligns with Reverse the Red's commitment to species recovery. Our team focuses on conservation action in 8 conservation hubs located in biodiverse regions all over the world, with our biggest center of activity being located here in the Southwest US. All of these hubs represent multiple species and ecosystems, dozens of partnerships, and decades-long investment in conservation and scientific advancement. Community relationships and sustainability are at the core of our projects in each of these locations, and where we look to support local priorities that align with species recovery. One of the tools that we're most excited about in terms of expanding globally is biodiversity banking. This is a tool we apply through our 6 sub-collections in what we call the San Diego Zoo Wildlife Alliance Wildlife Biodiversity Bank. These collections range from our native plant gene bank, which helps us restore native plants here in California, to our pathology archive that helps us diagnose and improve health outcomes for animals in our care. Perhaps our most famous collection is the Frozen Zoo, which contains the world's most diverse collection of wildlife gametes and cell lines, and has been used in amazing advances in conservation, ranging from generating over 100 genomes for vertebrate species to the conservation cloning of formerly extinct in the wild species, the Przewalski's horse and the black-footed ferret. We're proud to serve as an IUCN Center for Species Survival biodiversity banking, and we are working in partnership with the Animal Biobanking for Conservation Specialist Group to expand biobanking capacity in 4 target regions, including Kenya, Vietnam, Hawaii, and Peru. And just a shameless plug for our own species pledge, evidence of recovery success. The San Diego Zoo Wildlife Alliance has been working long-term in terms of conservation, breeding, and release with the Hawaiian crow, also known as the alala. This has been an initiative that has been in strong partnership with state, local, and federal partners. And a recent success that we would love to highlight is that in November 2024, 5 alala were released on the island of Maui, and they are still alive and well today and being monitored for their behavior. And so we can learn more about this release that can hopefully eventually inform the conservation outcomes that will be needed to take this species from being extinct in the wild to recovery. We're also committed to the future of the species' genetic diversity through a recent AZA grant to optimize biobanking protocols and expand on-island biobanking capacity for Hawaiian forest birds so we can support this species well into the future. Today in this webinar, we'll hear about we'll celebrate recovery across the Tree of Life from a fabulous panel of contributors. We'll start with Caitlin Brandt, who will tell us about the species impact and the species pledge for Reverse the Red. We'll also hear about turtles from Toby Thorne, fungi from Tamara Walton, amphibians from Muhammad Rais, and about the role of agave restoration and bat conservation from Kristen Lear. I'm very excited to hear about these talks, and I will pass it over now to Caitlin, and she can tell us more about the Reverse the Red pledge.
Hi, everyone, and thanks for the intro, Katie. I'm just going to share my screen really quick. All right, and then, oh, sorry, let's get it full screen. All right, so I just wanted to introduce the species pledge, the Reverse the Red species pledge today, and you've already heard Katie talk about the Crow Pledge, and you'll be hearing about more pledges from the other panelists. So what is the Species Pledge? This came from a need to figure out who was doing what species conservation where. We're all in our silos in conservation, and we don't always know what other people are doing. So we created this database, the Species Pledge, and it helps us with three things. It helps us find out what's happening where, what's not happening. So are there endangered, critically endangered species that aren't being worked on somewhere? And it also helps us increase collaboration between governments and conservation practitioners such as yourselves. You heard in the video at the beginning talking about the CBD Global Biodiversity Framework and Target 4. We're trying to connect those government initiatives, Target 4, to people doing work on the ground. So what is it really? It's a commitment to action aimed towards the recovery of wild populations. So it's a short form, 5-10 minutes, that captures your current or your planned work for a specific species. You can do a private pledge so we know who's doing what, or you can have a public pledge, which shows your organization name, the species you're working on, the country you're working in or countries, and the conservation actions you're doing for that halting extinctions Target 4. All of this helps us map global conservation action and align those efforts to halting extinction and recovering species. So why spend that time making a species pledge? It's part of that global contribution to Target 4. You get international recognition. The Reverse the Red Network showcases the work that people are doing. You've got that national alignment to work with your government. We also have increased collaboration in our network. You can request to meet another pledge maker, say they're an expert on something that you want to get into. We're trying to connect with each other across the world. And finally, it helps with access to funding. So we also, Megan mentioned at the beginning, the Accelerator Award. So the Reverse the Red Accelerator Award is 15,000 US, along with storytelling and strategic support to help a species that's endangered, critically endangered, or extinct in the wild. And that's opening again on March 2nd. So please check out the Reverse the Red social media to apply for that. All right, what makes a good pledge? So a strong pledge should commit you to actions that have measurable conservation impact. So I said a minute ago, we want to contribute to growing wild populations. If you're doing assessments, we want to increase knowledge to support their protection. It should involve monitoring. So even if you're not actually doing the monitoring yourself, still have that data to see what the distribution is. Are we reversing declines? What the total population is? Those kind of things. If it has a species recovery plan, your species, aligning that with existing strategies. And finally, addressing key threats. We want to tackle risks identified on the IUCN Red List to reverse the red. And I've got the example on the right here, which you're going to hear more about from Toby in a minute with the Blandings turtle. Okay, so exciting news this week is we're launching Pledge web app. So the Species Pledge has been around for over a year, but now there's a public database. So now you can search the pledge database to see who's working on the same species as you. You can edit your pledge if things have changed since you made your pledge. You can go in there and say, well, we're actually doing the recovery implementation now. You can also send that request to us, to me. You can email me at any time to connect with another pledge maker. And the website is pledge.reversethered.org, or there's a QR code here. So make a pledge today. We're also having a tutorial session on February 19th, which you can sign up through that QR code there, where I'm going to go through how to make a pledge if you want more information on how to do it. But I just wanted to say thank you for all the amazing work you do for species and for conservation. And again, email me if you have any questions about the pledge or the Accelerator Award. So thank you very much.
Thank you so much, Caitlin. And now, as was foretold, Toby Thorne from the Toronto Zoo will be joining us to talk more about their work with Blandings turtle.
Okay, yeah, good evening, everyone. It really a pleasure to be here in such illustrious company and to get to talk about some of the work that we're doing at Toronto Zoo. So I've already been introduced, but I included a quick intro slide here with a picture of myself in the field to prove that I still am, or used to be, at least a field biologist, and my background sort of primarily being in bats, but now broadening a little bit in this role at Toronto Zoo and getting to know some other taxa. And I want to change slides. There we go. And so, yeah, introducing our approach to conservation at the Toronto Zoo. We are a really conservation-focused zoo. Our mission as an institution is connecting people, animals, conservation science, and Indigenous knowledge to fight extinction. And doing that through collaborative science and through wildlife science is the major focus of our current strategic plan. And so as we sort of look to put that into practice, we're including knowledge and expertise from across the spectrum of the different sciences that exist at our zoo. And we're fortunate as a large institution to have experts in a wide variety of fields, including nutrition, a world-class veterinary team, reproductive science team. And so as we progress and as we move through this strategic plan, we're working hard to bring all of those pieces together. And a lot of the time in our history, we've had projects that so touch on maybe one or two of those, and where Reverse the Red has been a sort of help to us and a sort of addition to the work that we do is through pledges, is finding the ways to connect all of these pieces together. And so to date, Toronto Zoo has made 9 species pledges. We made 9 pledges last year, which was our sort of jump into this world of Reverse the Red and our commitment to that. And 8 of those were for Ontario bat species. And so, and with bats, it's sort of hard to work with just one, at least in the case of the bats in Ontario. So it's hard to work with one without really learning about and affecting the others. So it made sense to pledge for them all. And as you can see with the little yellow triangles there, 7 of the 8 species in the province of Ontario are now considered endangered. So there's a lot of work to be doing. And so our work with bats sort of ranges from a lot of monitoring to just to answer the basic questions of where they are, how they're using the landscape, and then working with partners to find ways to address the threats and the challenges that bats are facing. But I don't want to spend too long talking about bats, because I know Kristen's going to talk more about that with a really cool story about bats. So our 9th pledge so far is what I'm going to focus on, and that is the zoo's ongoing work with Blanding's turtle. So Blanding's turtle is one of the 8 turtle species in Ontario. It is endangered federally in Canada. It's at risk globally. It's a sort of really neat little turtle, also known as the turtle with the sun under its chin because of the yellow chin that it has. But it's a species that's facing a tough time, and it's a species that has been facing a tough time right in our backyard at Toronto Zoo, among other places. And so, and specifically within Rouge National Urban Park, which is Canada's, I think, currently only urban national park, and our neighbors and collaborators for a lot of conservation work at the zoo. And so Blanding's turtle is a species that the zoo has worked with for well over 14 years. And that initially began with working with the park, or the precursors to the park, in fact, to monitor the population in the Rouge watershed and to learn about how the population is doing there, and that identified the fact that this population was functionally extirpated, or there were fewer than 10 individuals and not a sustainable population of Blandings. And so back, say about 14, 15 years ago now, we committed to a project head starting Blandings. And so for head starting, we're not breeding them, but we're collecting eggs from the wild, and from turtles in a more stable source population. In fact, we collect eggs from the side of the road where they wouldn't be likely to survive anyway. And then we bring them back to the zoo, raise them for 2 years, at which point they are much larger than a wild 2-year-old would be. And we release them into the wetland. And so the first releases were almost 13 years ago. This year will be the 13th release. And so we've released over 700 Blandings turtles through this program over the 12 releases. And this program is designed in alignment with the federal recovery strategy for the species. So we're targeting both the short-term aim of stabilizing and maintaining Blandings turtle where it currently occurs, and then also targeting the long-term aim to increase abundance more broadly and to increase the number of populations that are out there, as well as ensuring they remain viable. And so to achieve that, when this program started, there were three original objectives. So one being to whether we could just reestablish the population in the Rouge. However, even if that works perfectly, the species is still endangered across its broader range. So we're interested to evaluate head starting as a broader tool, and then also having Blandings turtle, having this living laboratory in our backyard, using that to answer other questions about Blandings. And the plan was to do this for 20 years of releases to achieve a sustainable population, as you can see at the top there. And so we actually, almost exactly a year ago, we had an exciting meeting where we sat down and we checked in on this, and we really evaluated how things had been going. And so these were the original questions, some of the original questions when this project started, and we actually were able to sit down last year and say, we've answered some of these. So in particular, number 4, so understanding just whether the turtles survived and whether they could thrive in the wild. We now know at this point there are 12, 13-year-old turtles out there, they can. And taking that step of evaluation also allowed us to refocus our efforts and our resources onto some of the emerging questions. So we still don't know, we still haven't seen the turtles reproduce in the wild, but we know that's something that could happen any year now. So we're monitoring them closely. And we also revisited last year with an updated population viability analysis to look at our numbers and see whether we're on track to meet the original goal. And I think we're about 1 year off, which is close enough for me. And so things seem to be working quite well there. And the pledge, sort of, as well as connecting us with partners like Parks Canada, allowed us to, kind of to really to expand the beyond the park aspect to this conservation program. So we want to recover Blandings in the park, but we know we need to work beyond that. And so pledging the species is our step towards that. And finally, I'll just sort of finish with saying we're not done at 9 pledges. So I mentioned the bats, I mentioned the Blandings. We actually have a new pledge to launch and celebrate this year in association with Reverse the Red Day on Saturday. So I'm not going to say what it is, but hopefully you can all check out the zoo's social media and outreach, and you'll find out what that species is going to be. So thank you.
Thank you so much, Toby. That was excellent. And so moving from Canada to Australia, I would like to welcome Tamara Walton, who's the coordinator for FungiMap, who will be talking with us about some of the cutest colored fungi that you will see today.
Thanks very much. I'm hoping this works. So yes, I'm the National Coordinator for FungiMap, and I'm going to talk to you today about fungal conservation, or the lack of it, in Australia, and through the lens of tea tree fingers, which is not actually this fungi and not particularly attractive, and the role that FungiMap play through and run through our application to the Reverse the Red process. So you'll have to bear with me a little bit. I had some issues with my computer. Here we go. So despite their immense importance to humanity and ecology, fungi are largely overlooked in conservation in Australia and many other areas of the world, and often missed altogether in any discussion on biodiversity. Fungi represent around 9% of Australia's biodiversity, but under 12,000 fungal species have been described from a potential 250,000 species. Little is known about what fungi exist, their distribution, their conservation status, but we know that fungi underpin ecosystems and nutrient cycling. We have 36 Australian fungi on the IUCN Red List at the moment, and no fungi listed under Australia's Environmental Protection and Biodiversity Conservation Act as yet, which is our core Environmental Protection Act in Australia. So basically there's a huge knowledge and protection gap. So now this is why we have FungiMap and what we do. So we were established in 1995. We're a national not-for-profit citizen science organisation. And basically there are not enough mycologists in Australia, and there's currently no formal pathways for mycology either. So much of the knowledge that we have on fungi comes from citizen scientists. We map the fungal diversity via the FungiMap Australia project on iNaturalist, with the help of citizen scientists from around the country. We have something like over 125,000 observations at present. We also produce books like The Fungi Down Under, which is a key resource. We host webinars and do forays. And every year we hold a month-long Great Aussie Fungi Hunt. The last Great Aussie Fungi Hunt had over 32,000 observations from more than 3,500 participants. And to put that into context, that's 1 in every 7,662 people in Australia, with more than 1,300 species being observed. We also advocate for fungal conversation through education events, in the media, and with all levels of government. And part of this and part of the process that we do that with is by preparing a threatened species nomination for the tea tree fingers, which I'll talk about now. So as I said, not the prettiest fungi out there. It's endemic to Victoria. It's been on the listed as critically endangered since 2019. And that listing came in part from records collected by FungiMap and our citizen scientists. We worked with, and there's been no action on recovery since it was listed in Australia. We worked with experts and consulted with Victorian government and identified this as a great pilot species to actually explore how to do a threatened species nominations and achieve fungal conservation in Australia. So we worked with Sapphire McMullen Fisher, who's a renowned Australian mycologist, and prepared a nomination, as I said, to the EPBC Act. So with the listing, the nomination was a long and intensive process. I've got to say there was a lot of data and maps, and for a very small organisation like FungiMap, it required a lot of effort. But really, we see this as critical in terms of being a foundation for how government can both recognise fungi and actually implement recovery planning for the species that are most at risk. So I'll just talk a little bit about the process of our proposal to reverse the red. As I said, we wanted to use tea tree fingers as a pilot species for fungal conservation in Australia. And so this had a number of components to it. Firstly, we needed to increase the on-ground resources in FungiMap. As I said, I work 10 hours a week and everyone else is volunteers. We wanted to bring together the experts together in a workshop, key mycologists, citizen scientists, representatives from state and federal government, and identify what the current state of play was with regards to the conservation listings and actions for the critically endangered fungi that we have. This workshop was also planned to identify recovery group planning members. So the next step was to establish a recovery planning group, which would assist in us developing a recovery plan for tea tree fingers. It would also help us identify the next priority fungi species for nomination and listing, and then help us to actually develop a framework or a pathway, if you like, for fungal conservation in Australia. The other component of this was really about a field trial. So we wanted to not only have a recovery plan, but have additional information, given the lack of information we've currently got, additional information on on what would work for tea tree fingers. And so the field trial was basically aimed at better understanding how to maintain an ex situ population. There's only 13 known sites of this particular fungi species in Victoria, and that then would facilitate long-term genetic rescue. So where to from here? And this is where the pledge comes in, and obviously Tea Tree Fingers, we will continue to pledge to work towards actually developing a recovery plan and, you know, and actually using that as a pathway forward. So we will continue, FungiMap will continue to monitor and assist with the nomination assessment process. We're lucky enough that our nomination has now been listed as a priority by the Minister, a priority for assessment. We won't necessarily know where things end up until 2017. So we've still got a long way to go. We will continue to work towards the development of a pathway for fungal conservation in Australia, and we'll continue our work to educate, inform, and advocate. So really, we want to develop a collaborative framework on how to deliver fungi conservation and understanding across Australia. So we also run a small research grant round every year, which is funded by donations and memberships. We continue to seek commitment from all levels of government for the recognition of fungi as a separate entity to flora and fauna. It currently gets subsumed. And we will continue pushing for the Australian government to sign on to the Fungal Conservation Pledge at COP17. And that's really it for me. So first of all, I'd just like to give a couple of thanks. I'd like to thank Reverse the Red for giving me this opportunity today to share the work of FungiMap and the application process itself that we went through, because it allows small organisations like us to hone our skills and hopefully get better at it. I would also like to thank the amazing Steve Axford for the majority of the beautiful photographs I've used in the presentation. And thank you to all the citizen scientists, because without them, we would not exist. Our work would be just not happening, and our understanding of fungi would be greatly diminished. So I'd like to urge all of you to take a walk wherever you are, take some photos of fungi, upload them to iNaturalist, and you never know, you might even discover a new species. Thanks very much.
Thank you so much, Tamara, for sharing that. It's incredibly a huge responsibility to have to speak for such a large part of the Tree of Life. And I hope we can discuss that more in the Q&A. Next up, as we endeavor to cover as much of the globe and the Tree of Life as possible, is Dr. Mohammed Rais. He is an assistant professor at the Arid Agriculture University of Rawalpindi in Pakistan. Thank you so much for joining us today, Mohammed.
Thank you. Good morning, good afternoon, good evening, whatever time zone you are in. It's morning in Pakistan. Today, I'm gonna present very briefly about our journey, which started in 2014, and it is still going on. And I'm gonna present what we have done on endemic spiny frogs in Pakistan. Next, please. This journey started with the rediscovery of a frog from its type locality. Then we continued work and extended the foundational knowledge on this species. We proceeded towards identity and status confirmation. We studied niche suitability and distribution. We examined movement pattern, how this species and tadpoles move in its natural habitat. We conducted threat assessment, and at present we are doing work on gene flow, identification of population centers for the conservation of population of this frog species. Next, please. As you all are aware that of all vertebrates, amphibians constitute highest number of threatened species. Almost 41% of the world's amphibians, they are threatened with extinction due to a multitude of threats, including diseases, climate change. Next, please. Luckily, in Pakistan, we do not have any threatened amphibian species. But sadly, whatever we have in Pakistan, our amphibian fauna, it's not properly studied. We face problems. We have limited attention in terms of amphibians, amphibian fauna. They find less space in research, conservation, and management frameworks. There exist a considerable knowledge gaps in terms of natural history, population assessment, threat assessment, policy and management of amphibian fauna in Pakistan. We do have a few endemic species. These species are endemic to Hindu-Kush Himalaya, and of all these species, a few species, they are experiencing decline in the population. So the global population trend of these species show declining trend. Next, please. One of such example is Nenorana vicina, or Murray Hills frog. It's endemic to Himalayas, found up to the elevation of 3,000 meters above sea level. On the IUCN red list of threatened species, it is listed as least concern, but the global population trend is decreasing. Next, please. As I just said, we started this journey way back in 2014 when a group of our students collected specimens of frogs which we could not identify. And when we brought those samples to our lab and examined their morphology, we found out that those specimens belong to a species which was discovered from that particular region. And it was the type locality of this frog species from the Murray Hills. It was first discovered in 1872, and then due to less attention or less, you can say, a focus on amphibian fauna, people working on wildlife conservation, they did not carry out studies on the amphibian fauna of that particular region. So from that point onward, we started working on this species. Next, please. Although we did work on various aspects of natural history of this frog species, I'm gonna briefly discuss a few of them. These work were conducted by students from my lab. You can see we for the first time, one of our students, she gave a description of tadpoles of this species. It was not reported or published anywhere. So in 2020, he for the first time collected tadpoles from the type locality, and she gave morphometrics and diagnostic features of the tadpoles of this species, Murray Hills frog or Nenorana vicina. Then in the same year, we published findings on the occupancy of this species, and we found that elevation, permanent water bodies, and forest type, they were the predictor for this species. This species is associated in Pakistan with the subtropical pine forest and moist temperate forest, and some other forest type which are further up or north to moist temperate forest. Next, please. We for the first time provided the overview of amphibian fauna of Pakistan. We provided checklist, photographic guide to almost 21 amphibian species in 2021. including reporting the Mariel's frog. And in the subsequent years, for the first time, we provided genetic record of a few amphibian species like Minervaria peri. It was misidentified previously from Pakistan, and we also uploaded sequences of Nanorana vicina in the gene bank. Next, please. One of the exciting works conducted by another student from my lab, it was on the movement pattern through VHF radio transmitters. We put on BD2 Hollow Hill transmitters on Nanorana vicina and carried out a study on the movement pattern. And we found that they showed a high site fidelity. They were found within a small patch of stream. They were moving along the stream, not into the forest. That gave us idea that instead of focusing on a larger area, we should focus on microhabitat because these species, they did not exhibit much movement. Next, please. Then we mapped the niche suitability and distribution pattern of amphibian species which are found in our study area. We reside in Rawalpindi, Islamabad, and this is basically the foothills of Himalaya. And from here onward, we have Himalayan region. So here we have almost 9 amphibian species, including Mariel's frog. When we ran Maxent model, we found out that precipitation of warmest, coldest quarters, distance to rivers, and vegetation type, they were the primary environmental predictors of Nanorana vicina. Next, please. You can see the areas shown in mustard or yellow color. It's the northernmost part of Pakistan, and this part has more endemic species as compared to rest of Pakistan because Pakistan predominantly it's arid country. We don't have rainforest and thick forest area in the plain areas or in south. Rather, a mosaic of habitat in the north, which features different forest types such as subtropical pine forest, Himalayan moist forest, etc. These areas feature a few endemic species, including Nanorana vicina. Next, please. Next, please. Alongside doing the work on natural history fieldwork, we also carried out threat assessment, and we studied both climate as well as anthropogenic threats. In one of the studies by our student in the lab, we reared the tadpoles in our lab at different temperatures, and we found out that larvae of Nanorana vicina, they grew faster at higher temperature, but the body size was small. So high temperatures in this species, although supports faster metamorphosis, but it was making them unfit. Next, please. This is one of the recent work which we have just published. It's by 2 research students. In this work, we studied the impact of engineered nanoparticles, zinc and iron, on the growth and development of Nanorana vicina. And our results showed that any concentration over 10 milligram per liter, it delayed growth in the tadpoles of Mariel's frog. Next, please. Currently, one of our students, she is doing work on identification of gene flow pattern in different populations and metapopulations of Mariel's frog in one of the protected areas in the northern Pakistan. And so far, we found out that the most important population is found in the north western part, and this population needs to be given more attention in terms of conservation and management. Next, please. Just to give you the idea that although we don't have any threatened amphibian species, but as a researcher, as a student of herpetology, student of wildlife management, I believe that we should work on species when they are not threatened. It's the right time to study their natural history, to identify sort of threats they are facing and how they respond to those threats, so that we can proceed with the conservation efforts even when their population is not declining. So right now we are focusing on in terms of conservation and management, we are interested in habitat protection, climate adaptation legislation, which is lacking in case of amphibians of Pakistan, and surveillance of diseases, long-term mark recapture studies to better understand their movement pattern, and engaging the community and citizens for the conservation and management of amphibians. Next, please. In the end, I would like to thank Reverse the Red, Katie, Megan, all the speakers, participants, my institution, Arid Agriculture University, and my all other affiliations. Next, please. Last year, we had a series of activities on amphibian conservation. These are just the a few glimpses from our seminars and walk. Next, please. Next, please. So with this, I would end my presentation, and I'd be happy to answer if you have any question. Thank you.
Thank you so much, Mohammed. It's very inspiring to hear about all of the knowledge gaps you were able to fill for amphibians. And so in the interest of getting to our last and definitely not least speaker, Kristen, I am going to welcome Kristen Lear, the director of the Agave Restoration Initiative at Bat Conservation International, to share about her work. Thank you so much, Kristen.
Awesome. Thank you, everyone. Can you all see my screen yet?
Yes, that looks great.
Okay, perfect. So thank you so much. I'm excited to be here from Bat Conservation International and sharing about our recovery work for the endangered Mexican long-nosed bat. So a little background, BCI, we are a nonprofit that works around the world to end bat extinction. So we work globally on threatened and endangered bat species. And for today's talk, I'm focusing on this little bat, the Mexican long-nosed bat or greater long-nosed bat, Leptonicterus nivalis. Unfortunately, this species has undergone population declines of about 50% in the last few decades, primarily for two reasons: roost loss and disturbance of their roosting caves and their roosting mines where they roost throughout their binational corridor, and foraging habitat loss and fragmentation across a binational corridor that they migrate through. And this is hampered by their very slow reproductive rates. These bats, like most bats around the world, give birth to one baby per year. So recovery takes a long time to happen. And like I mentioned, this is also hampered by the fact that these bats are migrating very long distances, over 1,200 kilometers every year between Mexico and the Southwest United States. So you can imagine this is a lot of complicating factors that make recovery a long game. But we can measure progress not just in the number of bats present or species recovery, but in those incremental steps taken to get there. So we can measure progress in the number of partnerships established, recovery planning milestones, habitat protected and restored, the new knowledge and tools that we're developing to study the species, and increased public visibility and support. So I want to go through each of these and give some examples of what we've seen through our recovery efforts and the Agave Restoration Initiative here at BCI. So in terms of partnerships established and recovery milestones, in 2016, BCI helped found the Nivalis Conservation Network, which is a group of researchers on Nivalis, academics, government agencies, NGOs across the US and Mexico that are working towards conservation of the species. And we hosted our first meeting in 2016 of the network, and since then have been meeting periodically to help guide conservation and restoration efforts for the species. And as an outcome of this, in 2018, we published the US Species Status Assessment for Nivalis. And then in 2024, we published the Species Recovery Plan in the US. And those two documents provided the prioritization of recovery actions and provided a roadmap to delisting and to recovery. So importantly, in those plans, agave habitat loss across that binational migratory corridor was identified as a key threat to the species, and agave restoration as a priority recovery action. So in 2018, Bat Conservation International launched the Agave Restoration Initiative to restore that binational nectar corridor that these bats are relying on for their migration, feeding from these agave plants. And as an outcome, since 2018, we have, with local partners, planted over 185,000 agaves across that corridor, restored over 5,800 hectares of land. This is both wild land and managed lands with communities and farmers in both countries. We've supported 26 community and nonprofit nurseries to grow these agave plants and other locally important plants for restoration. We've launched 4 regional agave restoration networks to guide action at a more local scale. And we've partnered with over 100 partners to succeed and to accomplish this work. In terms of new tools and knowledge, I'm really excited to share about our eDNA, or environmental DNA tool, that we have developed over the past several years to detect these nectar-feeding bats from their food sources. So from agave plants shown here, or from anthropogenic food sources like hummingbird feeders. These bats leave a lot of spit behind while they're feeding, and they can, from that, we can detect the DNA and detect which species of nectar bat is feeding at those agaves or at those hummingbird feeders. And you can see in the right picture, swabbing agave flowers. It's a very, very easy thing to do. And this is actually the only non-invasive method for distinguishing the endangered Mexican long-nosed bat from its related species, the lesser long-nosed bat, Leptonicterus yerba-buena. So without having to capture these bats, disturb them, we can do these eDNA surveys across a large landscape, collecting a lot of samples, so it's much more efficient than traditional methods of monitoring. And as an outcome of this new eDNA approach, we have some exciting discoveries of range expansion or range shift areas of the Mexican long-nosed bat. So last year, we reported the first known recordings of the Mexican long-nosed bat in the state of Arizona. And tomorrow, we will be announcing a press release about another exciting discovery of the species from this eDNA work. And so these range expansion areas, this range information is now being used to update US, federal maps, as well as the IUCN range maps for the species, to make sure that we and development governments know where these bats actually are occurring. And so, for example, our federal partners are now using that new information to guide some wind energy siting in the US. So this is really important work that's helping guide recovery actions in the best way possible. And then finally, increased public visibility and support. This work with the Mexican long-nosed bat and agave restoration has really captured broader public attention, which of course brings with it increased public support and, importantly, funding for recovery actions. The Agave Restoration Initiative from BCI and our local partners has been featured in media such as Science Friday, the BBC. There was a BBC Storyworks Living Legacy series feature about the initiative. And all of these help garner public support for species recovery. And I think this is a really important part of the work. And I want to end just by saying that this is an all hands on deck endeavor. Working across a large binational migratory corridor requires partnerships. It requires all hands on deck, partnerships that drive this conservation success. So thank you to all of our local partners who are making this work possible. And here are, please follow BCI. Here are some links to our social media and some QR codes to our Agave Restoration Initiative page, and a page with some beautiful stories from local partners and videos of these bats and agaves and the interconnectedness of agaves and people and bats. So thank you all. I will leave this up for a second, but looking forward to questions.
Thank you so much, Kristen. And we are running a little short on time, so I welcome all of the panelists to turn on their cameras and join us for our abridged Q&A session here at the end. And so I guess one question that I have that kind of spans many of the types of projects that were discussed here is that many of the projects today focused on species or taxonomic groups that maybe aren't broadly understood in terms of their conservation need or their ecological importance. And so how has having a successful project been useful in, I guess, gathering more support for the work you do and communicating to decision makers that this work is worthy of support? And so maybe I can call on someone, or we can start with whoever wants to go first. Kristen, what about you? You work with bats, and do you ever have difficulty finding connections with the general public or decision makers about the importance of bat conservation?
That's a great question. Honestly, not much. I think bats are one of those can be polarizing topics that people either love or hate, or there are people in between. But even if they're not fans of bats, that's a connection. That's an in to have a conversation. And so in general, I don't think that's a hindrance. And with the Agave Restoration Initiative and these nectar bats, because agave plants themselves are so tied to history, they're tied to culture, they're tied to our economy with, products like tequila and mezcal, you have that other natural in to connect with people. So a person doesn't even have to care about bats or want to protect the bats, but they can still plant agaves or restore agaves on their agave fields or in their backyards and be contributing to bat conservation. But their motivation might be, tequila production. So I think that's a really great part of this story, is that you can come at it from different angles and gather support.
Thank you so much. And Tamara, what do you think about talking to government stakeholders about fungi? Do you feel like you have to inform them about the importance, or do you think in general there's a baseline understanding of why recovery is important?
So there is not a baseline understanding, I don't think, and many people don't understand fungi and where it fits, and they just see it subsumed as part of the flora when it's really not flora or fauna. It's a complete kingdom of its own, if you like. So there's that hurdle to get over, but I think one of the ways that we've approached that in terms of working with government and trying to build some level of support is really through the citizen science side of things. Everyone can go outside in their backyard, in their park, and look at fungi and take photos and get an idea of what's out there. I mean, that comes with the other side of things where people, worry about how it's going to affect their plant or if their pet's eaten it or all those sorts of things. But, it gives us an opportunity to at least engage on something that's out there so, you know, so readily and that so little is known about. Yeah. Thanks.
And Mohammed, you mentioned citizen science as a useful tool. How have you engaged people in Pakistan through citizen science to learn more about amphibians?
Actually, people who have some level of education or who are interested in wildlife conservation, we approach them and we encourage them to use tools like INET, and we request them to upload the sighting and occurrence records of whichever amphibian species they observe, because currently we lack any systematic data on the occurrence of many amphibian species. So it's one of the tools which we have available, and we ask our students and people who are interested in wildlife conservation to make use of it.
And Toby, how do you engage people with Blanding's turtle when the name Bland is so right there in the name?
Yeah, well, that's an excellent question. I think we don't have to try very hard to engage people with the turtles and a lot of the species that we work with. I think we benefit. One of the benefits of doing this work as part of a zoo is that we have a really big audience of folks that are interested to come out and see what we do, and also kind of in the sort of, as we think about the roles of zoos in modern conservation, are expecting to see those stories. And so Blandings, yeah, I guess there's bland right in the name. I think I prefer sort of the turtle with the sun under its chin as a little bit of a brighter and a more descriptive name that belongs to the turtle. And so, yeah, I think kind of engaging people with that works really well. And it's a nice, tangible story where we're putting an animal back into the wild, which is sort of easy to understand as a form of species recovery.
Well, excellent. Thank you, everyone, for sharing your stories with us today. And I'll kick it back to Megan to tell us more about what's going to be happening the rest of the week.
Yeah, we are so excited. I popped a few links into the chat. We have different conversations every day this week. So if you enjoyed this one, come back tomorrow. We have more species success stories tomorrow. We have a few other topics coming up as well. A look ahead at Reverse the Reds 2026 and a look back at what we did in 2025. Topic on storytelling, one about tech and collaboration, and a focus on national biodiversity strategies on Friday. And then on Saturday, there's at least one more conversation all about fungi. If tomorrow's presentation was your favorite part, come on Saturday. There's a full session all about fungi. And also happening on Saturday and throughout the week all around the world are satellite events. So take a look at that Reverse the Red Day page online. There's in-person events, there's other webinars organized by partners, lots of different topics, lots of different conversations happening, and also all across social media, all across press, we're amplifying stories of species success. So please take a look at that messaging, talk about with your comms team, sharing your own story, and please tag Reverse the Red. We would love to hear your story, like all of these stories, and we would love to amplify that and see even more of the amazing work that we're all doing to recover species. So thank you all so much again. Hope to see you again this week, and we'll celebrate all together online on Reverse the Red Day on Saturday. Thank you all so much.
Thank you so much.